Hello and welcome to another Mr. Carter Science Special. In this video we'll be looking at variation. If you take a look at the animals that you can see here on the screen, you'll notice that they are all very similar in many ways, but also very different in many ways. In fact, if you pause the video now, it won't take you long to be able to write down the list of the similarities these animals have and all the differences that they have. Well, the science word for all of these differences is variation, and I would imagine that your list of differences was much longer than your list of similarities. And that's because there is always a large amount of variation between different species. And here we have 9 completely different species, although all members of the animal kingdom. Now, if you take a look at these two animals and repeat that process, pause the video, write down the number of similarities and differences, then your number of differences is probably much smaller compared to the number of similarities. Both of these animals are, of course, dogs. They belong in the same species and therefore have much more similarities than they do compared to the number of differences. And there is always a smaller amount of variation between members of the same species. This is one of the ways that we can begin to recognize what a species is by looking at the variation that is contained in the differences between two individuals. If there's only a small number of differences, they're likely to be the same species. So what are the causes of this variation? Well, if you look at this picture of David Beckham here, you can see that he has many different characteristics. For example, his eye colour, whether he has tattoos or not, his skin colour, facial hair, the shape of his nose, the shape of his ears, his hair colour and his hairstyle. All of these things are characteristics that David Beckham has. But what has caused each of those characteristics? Well, we can try to take the characteristics that are listed here and to put them into 3 basic categories, because variation is always caused by either your genes, the genetic information in your DNA that you inherited from your mother and father and their sperm cells and egg cells, or the effect of the environment on your body, the effect of the sun, the UV rays, chemicals that might be in your makeup products, or in this case, bleaching David Beckham's hair, the amount of food that you have, how good your nutrition is, or in many cases, both. Many characteristics that you have or any animal has will be caused by a combination of both the genetics and the environment. Hair colour, for example, is genetic, but when we begin to modify it in the salon, we would consider that hair colour is caused by both your weight, your height, are also affected by both genetics and the environment. Your eye colour is solely genetic, unless you've gone and had expensive contact lenses or some kind of cosmetic surgery to change that. Things like your skin colour would be considered both, because although it's mainly genetic, if you go spend time in the sun, you may get a suntan or sunburn, which would affect the pigmentation in your skin. Your muscle size, again, would be considered that it is caused by both the genetics and the environment. If you have the genes for big muscles, you will naturally have larger muscles, but if you then go into the gym, you can change the size again and get even larger muscles. But some things like having a scar are solely environmental. You are not born with them. There is no genetic component to those at all. And in this case, David Beckham's tattoos would fit into that category as well. So scientists will often need to investigate variation, and in order to do that, you need to be able to make comparisons between how one factor has affected another factor in a living organism or a group of living organisms. This is never particularly easy because different living organisms will have different DNA, even if you use the same species. So in order for scientists to be able to investigate variation in plants, what they can use is they can use cloned plants, and this means that their DNA will be identical every single time. And to produce this, we can either use cuttings or tissue culture, as you can see here and here, in order to produce thousands of genetically identical plants that can then be utilized in your experiments to find out how an environmental factor can affect the phenotypes or the characteristics of a plant. And any variation between the different individual plants will therefore be due to that environmental factor and not the genetic factor, because you have created these plants to be genetically identical. In other words, their genotypes are all the same. However, in animals, this is much harder. It is unethical to go and create animals in large numbers that would be genetically identical or clones, and so scientists must rely on twins. Now, twins in animals can be used to investigate variety, because twins are genetically identical because they come from the same fertilized egg or zygote. Now, of course, we're talking about identical twins, not non-identical twins. However, it is unethical to separate twins and raise them in different environments. However, there are cases where twins have been accidentally separated and raised in different families, and this can help scientists in their research into variation in animals and into the effect of environmental factors on the phenotypes of different animals. If you're interested in this accidental separations, one of the best documentaries I have seen is on Netflix, and it's called 3 Identical Strangers. Now, twins look very similar and act in similar ways. However, scientists have measured different characteristics in twins, such as their IQ and their height and their weight, and these differences cannot be due to the genotype, so they must be caused by the environment. Now, this is a classic 6-marker question that you'll get with AQA, and it's asking you to design an investigation. So you can pause the video now, read the question, and give yourself 6 minutes and have a try. How well could you answer this question? You are given 20 pots containing identical cloned seedlings, all the same height and all the same colour. Suggest how you might investigate the effect of temperature on growth of these seedlings compared to the impact of their genes. Well, here's a model answer for you. And whenever you're thinking of these designing and investigation questions, you always have to be thinking of 3 things: your independent variable, your dependent variable, and your control variables. And finally, at the end, a statement about reliability. So in this investigation, the independent variable will be the temperature. And I will use 5 different temperatures: 10, 20, 30, 40, and 50 degrees. Always make certain you state which temperatures you're going to use, otherwise you will not get a mark for saying I will use different temperatures. It must be repeatable, so state what temperatures you're going to use. I will grow the plants with the same light intensity, the same volume of water, the same type and concentration of minerals in the soil. In this case, it wouldn't necessarily be the soil, but you might talk about a nutrient medium. These are the control variables that we're talking about here. So we've already talked about our independent variable and now the control variables. I will grow 4 different seedlings at each temperature so that I can calculate an average and improve my reliability. So that's our reliability statement. But we haven't talked about the dependent variable yet. What are we going to measure? Well, this final paragraph discusses that. The dependent variable. After 2 weeks, I will measure the height of each plant and compare it to the original height of a plant by calculating a percentage change in height. I will then be able to identify the optimum temperature to grow the seedlings at. The optimum temperature will be the temperature at which the seedlings have the greatest percentage change in height. Now we're looking at a percentage change in height because although we've been told all the seedlings began at identical heights, in the real world that's never quite possible. So we're interested more in how much has that height changed compared to the original starting height. Therefore we calculate a percentage change in height. So in this paragraph, we've mentioned the independent variable, the dependent variable. We've got our dependent variable discussed here, the control variables, all the things we're keeping the same, and we've got our reliability statement. So we should easily be able to get 6 out of 6 marks for discussing and designing this investigation. You can move the order of the paragraphs around to suit your own individual writing style. There's no problem with that, but you must always include all three variables and a statement of reliability. Well, I hope you've enjoyed this video on variation. I hope it's been useful for you. Let me know what you think of the video. Don't forget to like and subscribe, and reply in the comments below, and let me know what you've learned today. Thank you very much for your time.